Hello students, in the last video I think we have learnt about the osmotic pressure followed by tonicity, that is different solutions having various concentration that determines the direction of the diffusion of water molecules either into the cell or out of the cell. Children, I think we have learnt about the RBCs when placed in three different kinds of solutions, that is a isotonic solution, hypotonic solution followed by hypertonic solution. You observed the changes. Now in this video let us learn some more phenomena, or you can say the changes that occurs in the plant cell, which is different from what we have studied in the RBC. Okay, now let us start with the plant cell. Children, when you look at the plant cell, the unique feature of the plant cell, the plant cells have a cell wall along with the cell membrane. So when you look at the plant cell, children, the plant cell has a outermost rigid layer, that is the cell wall, made up of cellulose, which is the non-living membrane. So within the cell wall we find the cell membrane, which is the semi-permeable membrane, right? And then the plant cell has a large vacuole in which we find the cell sap, followed by nucleus usually is pushed towards the periphery, and we find the cytoplasm also pushed towards the periphery. Right, children? When the plant cell is placed in the hypotonic solution, or you can say that when a plant cell is placed in the normal fresh water or a tap water, we find that there is a osmosis occurring in the inward direction, right? So what happens in such case? We find that the cell membrane moves very close to the cell wall due to the continuous absorption of water. So you will find that there is a continuous inward movement of water molecules, right? That is the endosmosis occurs, due to which you will find a stage where the cell membrane moves very close to the cell wall, or you can say that after some time there is no more inward movement of water occurring. So there is no endosmosis occurring, where the cell reaches a condition where no more entry of water molecules occurs, even though there is a lot of water molecules in the hypotonic solution in the fresh water. So you can say that there is a development of osmotic pressure where it does not allow the movement of water molecules inside. Right? So there is an osmotic pressure developed. Fine, children. Now, what happens? So the vacuole also enlarges in such a way the size of the vacuole increases. Or in a simple word, I say the volume of the vacuole increases. The volume of the vacuole increases due to continuous absorption of water. Due to absorption of water. So compare the size. The size of the vacuole increases, right? And then, of course, the nucleus and the cytoplasm will be pushed towards the periphery, right? Okay, and this cell cannot accommodate any more water in it. Or just imagine your football. When you just look at your football, you have a leather covering outside. Within the leather covering, you find that bladder. Right? So when the bladder comes very close to the leather casing, where you cannot have any space between them, that is the way where the cell membrane and cell wall are in close contact with each other. So in this condition, along with the osmotic pressure, we have two more pressure developed. Okay, so there is a pressure exerted by the cell contents on the cell wall. So in such a cell, there is no more movement of water. We call this cell as a turgid cell. So what do we call that cell? As a turgid cell. Right? So in case of a turgid cell, the contents of the cell, that is vacuole, cytoplasm, all the cell organelles will exert a pressure on the cell wall. Okay, so that pressure which is exerted by the cell contents on the cell wall, right, we name it as a Turgor pressure. So how do we define the word Turgor pressure? Turgor pressure is the pressure exerted by, it's a pressure exerted by cell contents on cell wall. So don't write only the wall, it should be the cell wall. Right? At the same time, now the cell wall also exerts pressure on the cell contents in the opposite direction. So you can say that these two pressures are in such a balanced way which does not allow the cell to rupture out. So when the cell contents are exerting a pressure on the cell wall, at the same time the cell wall exerts a pressure on the cell contents. What do we call it? As a wall pressure. So how do we define the word wall pressure? It is nothing but the pressure exerted by cell wall on cell contents is known as a wall pressure. Children, so remember, Turgor pressure is the pressure exerted by the cell contents on the cell wall, whereas wall pressure is the pressure exerted by the cell wall. Don't miss out the word cell wall. Just don't write it as a wall. By cell wall on the cell contents, right? So this difference can be expected for 1 mark. So because of both the pressure, the cell is in the balanced state, right? There are certain conditions, especially in the winter season, okay? Sometimes when the Turgor pressure is more than the wall pressure, if the Turgor pressure exceeds or more than the wall pressure, okay, so if the contents are exerting so much of pressure on the cell wall, only in that condition the cell wall will rupture out and the contents are thrown out, which can be seen in certain fruits and vegetables, especially in the winter season. In that condition, what is the criteria? In that condition, the Turgor pressure exceeds the wall pressure. The Turgor pressure is more than the wall pressure. Only in that condition, the cell wall will rupture and the contents are thrown out, which can be expected for the give reason. Why do fruits and vegetables burst out? The reason is the Turgor pressure is more than the wall pressure. In that condition, the fruits and vegetables burst out. Right, children? So this is the condition where the cell has turned into a turgid condition, and it has turned into a state known as a turgid state, and the condition we call it as a turgidity. What do we call the condition as? Turgidity. Right, children? So how do we define the word turgidity? When a cell attains the turgid state. Okay, so turgidity, let us learn the definition. So turgidity is a condition. It's a condition in which the cell wall is rigid. Cell wall is rigid and stretched by an increase in the volume of vacuoles due to the absorption of water. Children, look at the definition. Only if you understand the meaning of a turgid condition, then you can put forth in a proper way. Yes? So what is a turgidity? So it's a condition in which the cell wall is rigid. So it means the cell wall will not rupture. It becomes rigid and it is stretched. So it moves out. Right? In such a way that the cell membrane and cell wall are very close to each other. So that is a condition of a cell wall. So in a turgidity condition, the cell wall is rigid and stretched. It is almost stretched. Why it is stretched? By an increase in the volume of the vacuole. So there is an increase in the volume of the vacuole. Why there is an increase in the volume of the vacuole? This is due to the absorption of water, due to the endosmosis. So I repeat, turgidity is a condition in which the cell wall is rigid and stretched by an increase in the volume of the vacuole due to the absorption of water. Fine, children. Now, let us place this turgid cell in the salt water. right? So when you place the turgid cell in the saline water, right? So what is the change that we observe? Right, children? Let us see. Right? This is as usual cell wall. Fine. Yes. The second layer is the cell membrane. Fine. The vacuole. Right. You will have a nucleus followed by cytoplasm. So this is the nucleus followed by cytoplasm. Children, just compare what is that we have learnt in the earlier diagram and what is that we are learning now. We call this cell entering into a flaccid state, right? And the condition is known as a flaccidity. Observe, in the earlier case, the cell membrane and cell wall were very close to each other. The size of the vacuole is so much, right? Look at this condition of the cell. The minute when the turgid cell is placed in the saline water, the cell membrane will withdraw itself from the cell wall. The size of the vacuole decreases. Cytoplasm also loses water. Okay, this condition is known as a flaccidity. So, how do we define the word flaccidity? Flaccidity is a condition in which flaccid cell is a state of the cell. Flaccidity is a condition. So, flaccidity is a condition in which the cell is no more tight, or you can say that the cell content is shrunken, right? So you can say that the cell is no more tight, no more the cell wall and cell membrane are very close to each other. So it has lost that turgidity. So it is no more tight. The cell membrane has withdrawn from the cell wall. The size of the vacuole has decreased. The cytoplasm has decreased in its volume. So such a condition we call it as a flaccidity. So what is a flaccidity? It's a condition in which the cell is no more tight or the cell content is shrunken. Children, if this continues for some more time, next the cell attains a one more condition known as a plasmolized condition or a plasmolysis. So what happens in the plasmolysis? Children, observe the change. Yes. As usual, cell wall, right? Come on. This is the cell membrane. Look at the cell membrane in the earlier diagram and here, right? Then so you can find the vacuole, but it is a shrunken vacuole. Right? So we find the nucleus, we find the cytoplasm. So we call this condition as a plasmolysis. Look at the difference between a flaccidity and a plasmolysis. Totally the cell membrane has withdrawn from the cell wall. The vacuole totally shrunken because there is a loss of water from the entire cell. Why there is a loss of water? Because the cell has been placed in the saline solution or in the salt solution. So it means the cell is coming in contact with the hypertonic solution, the solution which has a more strength, which has more number of solutes. So the cell is in contact with the hypertonic solution, hence the cell is losing water. Right? So how do we define the word plasmolysis? So plasmolysis is the contraction of cytoplasm. Yes, children. So it is the contraction of the cytoplasm from the cell wall caused due to withdrawal of water when placed in hypertonic solution. Yes, children. So how do we define the word plasmolysis? So it is the contraction of cytoplasm. So it means it is referring to both cell membrane along with the cellular contents. So plasmolysis is defined as the contraction of cytoplasm from the cell wall. Why? Because there is a withdrawal of water from where? From the cytoplasm, from the vacuole. Why is it so? Because the cell is placed in the hypertonic solution. Children, so how do we define the word plasmolysis? Plasmolysis is defined as the contraction of cytoplasm from the cell wall due to the withdrawal of water when the cell is placed in the hypertonic solution. Right, children? Okay, now this is a diagram which can be expected for about 2 marks. They'll ask you to draw a diagram of a plasmolyzed cell. You have to draw a cell wall, shrunken cell membrane, and especially you should mention about the shrunken vacuole. And children, can you guess what do we find in this space? The space between the cell wall and cell membrane, what do we find? Do you find the air? No. The cell is placed in the hypertonic solution. So definitely the space between the cell wall and the cell membrane, we find the salt solution or we find the sugar solution, in whichever the solution the cell is placed. So the space between the cell wall and the cell membrane, we find the hypertonic solution. So sometimes they will ask you to draw the diagram of a plasmolyzed cell. Okay, you have to label all these parts, and especially you should draw you should label the shrunken vacuole, the cell membrane properly, the cell wall properly. Right? Now, children, can we have this plasmolyzed cell coming back to the normal condition? Can we have a turgid condition in case of this plasmolyzed cell? Yes, it can be attained. It can be obtained. How is it? So when a plasmolyzed cell is placed in the, when the plasmolyzed cell is placed in the hypotonic solution, again place it inside the hypotonic solution, put it back to the fresh water. So when you put back the cell back into the fresh water, again you will find the cell membrane moving towards the cell wall. The size of the vacuole wall increases due to the absorption of water. Right? The cell moves back to the turgid condition, and that reversal of plasmolysis, or the cell coming back to the normal turgid condition, we call it as a deplasmolysis. We call this condition as a deplasmolysis. Right, children? So what is the meaning of a deplasmolysis? When a plasmolyzed cell is placed in the fresh water or in the hypotonic solution, the plasmolyzed cell turns into a turgid cell. So this reversal of a plasmolysis is known as a deplasmolysis. So if this cell does not receive any more water, it may lead to the death of that plant cell. For some period of a time, okay, the leaf or the plant cells, they wait till they absorb the water. When there is no more availability of water for a long period of a time, in such condition only the plant cells die, if there is no water provided to the cells. Otherwise, they remain in the plasmolyzed condition for some time. The minute when they receive the water, they become, or they change into a deplasmolyzed condition and continue to survive. Fine, children? Okay, so these were the three phenomenon observed in the plant cells, such as turgidity, flaccidity and plasmolysis. Children, the definitions must be learnt as it is. So understand the concept and then you can define the words easily. So use the proper words and then you can write the definitions. Is it clear, children? Okay, now based on this, you can have a number of give reasons. You can have a number of give reasons based on this phenomenon that I have taught you. Okay? Let me take up one simple example. Now, whenever you have a throat infection, right, children? Okay? So, whenever you have a throat infection, yes, what is the remedy? At home, Mama will ask you to gargle the saline water. So, what is the meaning of gargling the saline water? You're going to just take the salt water and then hold the salt water in your throat and then try to, vibrate that saline water. Okay, when you do this twice or thrice, you can experience that the pain reduces. Can you guess why? This can be one of the give reason question asked. Can you guess? Fine. Children, in the throat you have the pain because there is a growth of bacteria in the throat. Right? So when you gargle the saline water, so it means the saline water acts as a hypertonic solution. The saline water acts as a hypertonic solution. We know that when the cell comes in contact with the hypertonic solution, what happens? There is an exosmosis. There is an outward movement of water from the bacteria into the surrounding solution. So there is an exosmosis occurring, and what happens to the bacteria? The bacteria attains the plasmolized condition. So bacteria attains a plasmolized condition, due to which there is a death of bacteria. All the cells of the bacteria will be destroyed because of the saline solution, which acts as a hypertonic solution, and due to which there is an exosmosis from the bacterial cell, and the cells of the bacteria, they attain the plasmolysis condition, due to which your pain will reduce after some time. So when they ask you such reasons, children, you have to be alert. You should say about the type of the solution, right? You should say which direction the osmosis has occurred and what has happened to the cell. Okay, that is a hypertonic solution followed by exosmosis, and which leads to the plasmolysis condition. Children, let us recall about the potato, right? When a potato is placed in the fresh water, it remains fresh for a long time. Why is it so? Can you guess? Because the water behaves as a hypertonic solution, right? So there is a endosmosis occurring, due to which the cells of the parenchyma remain turgid. So what do we call it as? The cells of the potato remain fresh because of these three phenomena. Again, you should explain about the type of a solution in which the potato is in contact, that is a hypertonic solution, right? And then in which direction the osmosis occurs, that is an endosmosis, and the cells remain fresh or the cells remain turgid. Okay, like this they can ask you a number of examples, but remember that you are going to use such keywords when you are writing the reasons. Just look at the concentration of a solution surrounding the cell, and then based on that you try to answer. Fine, children? So go through the textbook, learn these definitions thoroughly, then it becomes easy. And especially you should practice the diagrams of a turgidity condition followed by the plasmolysis condition, which can be expected for about 2 marks. Right? Okay? Yes, children. Thank you.